Alright, so welcome to the last lecture of the semester. This one, we're gonna keep this one simple. We're gonna keep it short. I'd like to go into more detail on this, but just given the time and everything else that's going on this semester, we're gonna trim this one down to the bare essentials. I wish we had more time to talk about these things in person. I wish we had more time to talk about HIV in person. The last lab I worked in during my postdoc, I worked on HIV entry, and so it'd be really great to talk to you guys more about these things in detail. But wishes for horses. Let's move on. All right. Prions and viroids. These are the cool, freaky things in virology that aren't technically viruses. And they're some of my favorite things to talk about because they're weird, freaky things. But again, just timing and so forth. So prions, that actually comes from a weird attempt at an acronym. They're infectious proteins. And these, again, are not viruses because there's no nucleic acid involved at all. But getting to that point and understanding that prions are just infectious proteins and there's no nucleic acids, this was controversial for a really long time. There's actually still a couple holdouts that insist, no, there must be nucleic acid somewhere, we just haven't found it yet. And who knows, maybe they could be right. One of your online assignments is about this. Your practice essay is looking at the evidence or looking at the arguments both ways. Can proteins on their own really be infectious? And these proteins are the cause of transmissible spongiform encephalopathy. All right, we're going for complete, not perfect here. That's my mantra for the rest of the semester, and should be yours as well. Spongiform, because your brain looks like a sponge afterwards. Like, there are all these plaques are formed in your brain, and your brains essentially look like Swiss cheese. These are always fatal. They're very rare, but they're always fatal. All right, and then we've got viroids, and viroids are also not viruses, right? That they don't have a capsid. There's no protein capsid. They're infectious RNAs, and these are diseases of plants. Remember, though, when we talk about these at the end of this lecture, that hepatitis D bears some resemblance to these viroids. Okay, so human prion diseases, or prions. I've heard it pronounced both ways, and because we're not together, it doesn't matter how you pronounce it, because I can't hear you. So prion diseases or prion diseases. We've got this nice table here. The first one that we learned about was Kuru, and this is a result of cannibalism. And this is not cannibalism from a crappy B horror movie, but this is cannibalism of deceased relatives. And there's some good links on Canvas. Under the extra study materials, there's a good article about this, an NPR article, and also there's the movie link on Canvas to watch. But basically, people felt that if you're dead and buried, then it's going to be like the worms and the maggots and the other insects and stuff that consume the dead body, and shouldn't instead it be consumed by people that loved the person. And so the families ate the bodies. And then, kind of by random chance, one of the people had a misfolded protein, and then that led to their death. And then, when people consumed their body, that misfolded protein spread to them, and then spread to other people, and spread to other people. and this was really decimating the South 4 people. And once they kind of figured out what was going on, that this was being passed on through the cannibalism, they stopped that process. They stopped it, but it has such a long incubation period that even though they've stopped this decades ago, they still have a couple people coming down with this now. So we've also got Creutzfeldt-Jakob disease. We'll talk about that. These prion diseases has a really long incubation period. All of them have a very long incubation period, with the exception of the V that stands for variable or variant, the variant Creutzfeldt-Jakob disease. But all the others have a incubation period, sometimes of decades. And then after the signs and symptoms appear, death occurs. And then that time can vary. So with Kubru, within 6 months, some of them a little bit longer. So the incubation period for most of them is very long, and then death after initial symptoms appear usually occurs within a few months. So the initial symptoms are going to be an unsteady gait. You might have problems with your voice, tremors, and then patients have problems walking. So then you have the sedentary stage where people can't walk anymore. Kubru was actually called the laughing disease. That people lose control of their emotions, and so you might have like random outbursts of laughter. But there could also be, depression, the other extreme too. And then eventually, people can't sit up anymore. They can't speak anymore, and then they pass away. So this was discovered in the 70s, and prion stands for proteinaceous infectious particle, or PRP. And it's still, it was at the time just groundbreaking, like how could you possibly have an infectious protein? And there are still some people that argue we haven't found the hereditary nucleic acid component to it. They've also found that these prions are dangerous because they're really resistant to decontamination. It's a misfolded protein, so heating the misfolded protein doesn't really cause it to misfold more. It's really, really difficult to sterilize equipment. There have been cases, even locally in Maine, where there possibly contaminated people in hospitals because of prions that they couldn't get off of surgical equipment. So they say if you have undiluted bleach, and you let it soak in undiluted bleach, and then you have to autoclave it for almost 5 hours to properly decontaminate materials. So normal autoclaving procedures, you don't have to pre-treat in bleach, and you normally autoclave for 30 minutes or an hour tops. So here we're talking about something 5 times that. So these are really, really stable, really hard to get rid of. All right, so there have been a lot of questions. The normal folded cellular prion protein, like what does it do? When it misfolds, it kills you, but what does it normally do in the body? And there have been a couple different theories about it, but one of them is that it's really important for binding up copper, and it's important for reducing oxidative stress, and it's important for protection against copper toxicity. But when it misfolds, Then one misfolded protein can bind to a normal protein and it causes that protein to misfold. And then you've got two misfolded proteins and they can bind to other normal proteins and cause them to misfold. And then you get this accumulation of fibrils. So these proteins combined and they accumulate, they polymerize. So prion proteins are like ice 9. So for those of you guys who are fans of Kurt Vonnegut, in The Cat's Cradle, there was this molecule called ice 9 that was ice. It was water, but it was ice solid at a room temperature, and you could use it like if the military was, if you were trying to march across like a swampy place, you could put some in the water, like put the seed molecule in, and then all the water molecules that it touched would also become solid, and so you could use it to cross a swamp. But all water is kind of connected to all water on the planet, and so that kind of destroys the planet. So prions are like ice 9 put in your brain. So you get infected with one infectious protein, and it infects another, then another, then another, then another, then another, and then your brain turns to Swiss cheese. So here is a cartoon illustrating, you've got your normal folded protein right here, and you've got these alpha helices and so forth, and then when it's misfolded, it has more beta sheets in it. And then the misfolded protein, they clump together, they form these aggregates, or fibrils, that cause the cell to die. So they form these big plaques, and your brain turns to a spongiform encephalopathy. Encephalopathy. Goodness, it's the end of the semester, and I'm getting tongue-tied. So here's an image of the pathology. Different types of prions are going to target different areas of the brain. So Kuru here, Creutzfeldt-Jacobs up here, or up here. So different types of prions will target different areas of the brain, but they all turn your brain to Swiss cheese where they infect. So how does this get into your brain? It travels up the nerve. So you get this through ingestion, right? You're eating contaminated food. You get this through ingestion, travels up the nerve to the brain. Or may travel from the hypoglossal nerve from the tongue to the brain. We don't know how much protein is what the infectious dose is, right? So we don't know if you need to be exposed to 5 prions or 10 prions or 1,000 prions. We don't know what the infectious dose in people is. We do know that the infectious dose in animals is very, very, very low. So most people are going to get it through diet. So this could be through cannibalism, or this could be through eating other animals that have been affected with this. So bovine spongiform encephalopathy, or mad cow disease, right? So the prion responsible for mad cow disease is associated here with the Creutzfeldt-Jakob disease. Also, perhaps through a botched medical procedure, so contaminated surgical tools, for example, corneal grafts from an infected donor. Maybe they don't know that they have it. Duramatographs. So this is again pretty rare, especially in the States, but if you do get it, it's fatal. So we've got the infection version, and then we've got the inherited version. So there are a few people who've inherited mutations that that lead to Creutzfeldt-Jacobs disease, or fatal familial insomnia. So there's a few inherited ones. So most people that get this, the classic version is a very long incubation period, and most people die 68 years of age. But the variant form, people were dying really, really young. And they found that these people who have the variant version, they have a couple mutations in the protein that makes them more susceptible to this. We've already kind of gone through the signs and symptoms. So we start off with some mood changes. Remember, Kuru was the laughing disease, anxiety, and then the more neurologic symptoms. So you have spasms, you have problems with motor control, and then you lose the ability for speech. You lose the ability to really move, to walk around, to even sit up, you go into coma, and you die. Now, one thing just to point out here is a lot of these symptoms are associated with other neurodegenerative diseases. So it's exceedingly difficult to diagnose neurological conditions anyway. But here it's really hard to diagnose, and you never know for certain you have this until autopsy. And they cut open your brain and see what it looks like. So there are different mutations in this gene that we normally have, and we know that some of these mutations make you more susceptible, especially for the variant version. And honestly, I'm not going to ask you to memorize, guanine versus adenine. I just want you to be aware that different people have a different genetic susceptibility to this. So, the species barrier, right? So this is supposed to be mad cow disease. What's going on with infecting people? So this is a pretty conserved protein, and if it misfolds in the cow, the proteins are similar enough that this can also lead to disease in people. And so where did cows get it? Cows got it from sheep and other cows. So a common way of adding protein to cow feed is ground up leftover meat, bones, corpses. So the leftover animal material is ground up and added as a supplement to cattle feed. And so the cows were essentially practicing cannibalism, or eating sheep, which I suppose isn't cannibalism. But anyway, so they think that scrapie, which has been in sheep and goats for a long time, was around first, and then this got introduced into cows, and then up the food chain into people. Now, it's called scrapie because in this version, this prion disease, part of the change in behavior and the first part of the disease symptoms, these sheeps and goats, they scrape themselves against anything they can, fence posts or trees or what have you, and they scrape off all the fur. They even scrape off their skin until they go raw, and then when they die shortly after. But this practice of using basically the waste animal parts as a supplement feed for cows now has been banned. So they don't do this anymore, but they think this is how it initially spread from the sheep and goats into cows, and then from cows into people. So this was first noticed in England, and after they started figuring out what was going on, they banned import of British beef and British meat products into the States and into the rest of Europe, and England killed a lot of cows that they thought might be infected, and the chance of us getting this in the States now, or the rest of Europe, is pretty low, but it's not zero. And in England, they've done a lot of things to try to prevent the spread of this. So we think that in the future, the incidence of this in England might be going down. So this is still pretty rare, but it's not, the chance of you getting it's not zero, it's just very low. So as of 2015, they had 177 deaths since the start. So so between 94 and 2015, they had 177 deaths. So definitely a problem, but hopefully will not be increasing too much more. Hopefully we've got a handle on it now. The big question mark, right? The big problem is the long incubation period. So is it possible that a lot of other people in England, or even people here or in Europe, were exposed to this before the bans on British meat, before the England kind of had this sorted out, and in 20 years we'll get sick? Maybe. We don't really know. That long incubation period is the problem. There are two main types or strains. The first was the one that they identified in England. They found another one here. It's very likely that this may have arisen spontaneously. Again, they have now banned the practice of using the ground up waste animals as a protein supplement. So hopefully we have avoided more cases in the future. Now, another concern that we have, chronic wasting disease. So we know that scrapie went from sheep and goats to cows and then to people. Here in the States we have chronic wasting disease. Is it possible that this can move to people from the moose and the elk? We don't know. Maybe, right? So there's a big question mark that we have here. Can this move into people? Can this cross the species barrier? So what this does in moose and elk and deer is it leads to wasting away. So you can see here a deer that's near the end of its life. This is found in free-range deer. This is also found in domesticated or captive deer herds. So people like to eat deer. Venison tastes good. So what are we going to do about this? Is this a problem for people? Again, we don't know. One of the slightly concerning things is this doesn't have to be spread through cannibalism, or this doesn't have to be spread through, elk eating dead elk, but contaminated materials can get into the environment and can be taken up by plants and then can be spread to new animals. Right, so this uptake from the soil is of particular concern here. Can this spread to people? again, we don't know. The only way to really know for certain would be to take this and inject it into people and see if they get sick, and clearly we can't do that. That would not be ethical. So looking at other species barriers, looking at ferrets. If you feed them ferrets, they were not susceptible, but if you injected into their brain, they were. If we look at mice, and these are mice that have been engineered to express the human prion protein, they were not susceptible. But that's not a perfect model for people because it's still a mouse brain. A couple different studies have shown here in animals different types of monkeys. So squirrel monkeys who were exposed either by injection or by feeding were susceptible, but other species of monkeys were not. Here, these studies were done in a test tube, where they showed that the chronic wasting disease prion could, in tissue culture, convert the healthy human protein to the misfolded prion version. So again, we don't know. Testing for this, you can test the animals. Again, you really kind of have to slice open their brain. So there isn't a great way to test for this. If you are a hunter, and I'm not against hunting, but if you're hunting, you need to take some recommended precautions. Field dress the animal, leave the most infectious elements behind. In other words, debone the meat, leave the spine especially, leave the skull especially, because those are the most contagious. We're going to end with a very brief discussion of viroids, or viroids. So viroids infect plants. These, remember, don't have a capsid. It's only RNA, which is kind of crazy. These are not double-stranded. It's one piece of RNA that is folded back on itself. So if we had more time, and if we were in person, this would be a really great opportunity to introduce you guys to the field of RNA chemistry. And RNA can do all sorts of amazing things, and it can form all kinds of really, really cool secondary structures, and they can take on catalytic activity. They can even take on autocatalytic activity. RNA is really, really cool. You could have your whole career just studying different forms of RNA, but alas, we're going to keep it relatively simple here. So we've got two big categories here of viroids. The first are going to accumulate in chloroplasts, and they have a quasi-rod-like structure. So you can see they have these branches here. Here's a couple branches. Here's some pretty extensive branching. Alright, then we have the Hospiviroidae, and these form a true rod. So there's no branching here, and they locate in the nucleus. It's important to note, these are not viruses. Remember, most RNA viruses are going to replicate in the cytoplasm, but these do not replicate in the cytoplasm. They replicate either in the nucleus or in the chloroplast. Alright, these viruses, again, they're not viruses, they're not viruses, they're not viruses. Say that 10 times fast. These viroids are only that RNA genome, so they are not bringing in their own RNA-dependent RNA polymerase. They hijack the host RNA polymerase, again, which is kind of crazy. And remember, this is also kind of similar to the hepatitis D virus. And, like hepatitis D, they also replicate using rolling circle replication. and they have an autocatalysis here. So after you replicate the genome, you get this really long strand that's got like multiple copies, and the RNA can fold upon itself and self-cleave itself. So it has that ribozyme activity to self-cleave itself. And then after that cleavage, you get the circularization where, and again, it's not really like a circle like this when it's done. It folds and makes these cool secondary structures. So like it does like close the loop, but it's got all of this complementary binding to get these cool structures. So it's just easier to draw it like a circle. Okay, and a couple examples of these viroids. They can infect, there's several examples in plants. I'm just going to highlight these two, but this is common in tomatoes, potatoes. This is the first one that was characterized in potatoes. So you've got normal tubers and then the ones that were infected. Here's a picture from this paper. This is actually a really big concern for chrysanthemums. And a lot of people, when they're going into research and they go into molecular biology or virology or microbiology, they're like, I'm going to cure cancer, or I'm going to, cure HIV, or I'm going to cure Mycobacterium tuberculosis, or some other human disease. Lots and lots of research is human-focused, because, we're people. But plant viruses and plant viroids and plant bacteria and plant fungal infections are also really important. They can be super duper important in the environment. Looking at pine rust is a really big thing ecologically. Tobacco mosaic virus. Lots of plant things are important too. And so it's not just important for the environment, but also economically. Lots of people like to buy chrysanthemums. All over the world, people like to buy chrysanthemums. They're pretty. I have some in my garden. They're pretty. I don't know what else to say about chrysanthemums. I'm not really a plant person, other than plants are pretty. And I have flowers and fruit trees in my garden. My point is, and I know you can tell I'm obviously going crazy at the end of the semester, but economically, plants are really, really important. And so disease in chrysanthemums, so like here's a healthy flower, and then these are ones that have been infected. People don't want to buy sickly plants. And so here you can see like there's two healthy ones here, and then the two sick ones right here. So these are the sick ones. So this is a stunting of growth, and it causes the misshapen flowers. This is how they're detecting it on the gel. But this causes lots and lots of damage economically to the agricultural industry when you get an infection in produce that we eat or flowers like chrysanthemums. All right, this is it. This is the last of the lecture material for virology in spring of 2020. You've made it to the end. Congratulations. I hope everybody at home is doing okay. Thanks for sticking it out this long on this crazy semester. And please send me an email or let me know in Canvas chat if you've got questions.